Hello everybody. In this video we are going to be looking at what is called mitosis. Now mitosis is when diploid cells basically clone each other. And so this would basically be a body cell, it could be a fungus cell, it could be a plant cell. It's pretty much anything that's not an egg or a sperm. So when you cut yourself, you need to repair and make new skin cells. Or as you grow, you need to create more cells. And so our bodies do this through a process that we call mitosis. Now, just as a review, remember diploid cells mean that they have both parts of the recipe. If you think of DNA as a cookbook, and cookbooks have recipes in them, you have two parts to each recipe, the ingredients part and the instructions part. And you have to have both parts of that recipe to create that protein. So likewise, when we look at our genes, we have to have two parts, and we have to have both of those parts in order to create the recipe, which is the protein. And so diploid cells contain, in humans, for example, 46 chromosomes. So dye lets you know that they have both parts of the recipe, and therefore they're able to create proteins. Now, in the previous video, we talked about the cell cycle. And the cell cycle is basically what we consider the normal lifespan of the cell, from the time it's made to the time it either dies or goes through cell division. And so in the case of the cell cycle is where the cell spends most of its time. Like 99.999999% of the time, the cell is just living its life. Well, some cells, like nerve cells, never go through cell division. They cannot repair themselves. We cannot make new ones after a certain point in time. And so we take the cell cycle and we actually divide it into three different phases, what we call interphase, mitosis, and cytokinesis. Now, you will see that mitosis and cytokinesis often go hand in hand, and they're often called the M phase, M for mitosis. Okay. So when we talk about the cell cycle is basically the life of the cell. Now, most of the time, the cell is just living its life, doing whatever it is it's supposed to be doing. If it's a plant cell, it's doing its plant cell jobs. If it's a brain cell, it's doing its brain cell job. What we call the normal life of the cell is this thing here called interphase. Okay. Interphase is the cell living its life. So like I said, 99.99999% of the time, the cell is in interphase. That's when the cell is doing whatever it is it's supposed to be doing. Things like nerve cells, after a certain point in time, never leave interphase. They will never go through cell division. Okay, you know that when nervous cells are damaged, they cannot repair themselves, they cannot replace themselves. This is where they're kind of stuck. So when we talk about mitosis or cytokinesis, this is when the cell is actually going through cell division. So most of the time, the cell is sitting in what we call interphase. Inter meaning between, phases mean the phases of mitosis. So when we talk about the cell cycle, interphase is part of the cell cycle, but then we can have what we call mitosis, and that's where the cell actually goes through cell division. So like I mentioned, interphase is when the cell is doing its thing. It is living its life. It's doing whatever it is that it's supposed to do. However, if it realizes it needs to divide, so contact inhibition is lost or it's gotten too big, we have these three steps. Okay. Please note that these three steps only happen if the cell needs to divide. So for example, nerve cells will never go through these three steps. Okay. But interphase, the cell is living its normal life. If the cell realizes it needs to divide, it will do these three steps. And these three steps will be at what we call the end of interphase, and these three steps will trigger the start of what we call mitosis. So if contact inhibition is lost, so the cell has cut itself, or your body has sent hormones to your growth plate, and cartilage cells have died, it says, okay, bone cells, you need to go through mitosis in order to make more bone cells so that our bones can get longer. You know, that's how you grew, that's how your hands get bigger, that's how your feet get bigger. So in the case of interphase, if the cell realizes I need to divide, the first thing it will do is what we call the G1 phase. G1 stands for growth, and this is where it will grow to almost twice its normal size. Now, once it starts this growth phase, it has committed itself. Because if it goes to twice its normal size, we have to worry about DNA overload. We have to worry about surface area to volume ratio. These were things that I talked about in my other video. Why will cells need to divide? Okay. So it's going to grow to twice its normal size, because when you go through mitosis, you're making clones. So if the first cell is the size of a dime, the cells it makes has to be the size of the dime. So they have to double in size so it can split evenly. During this process, you'll also have what we call the S phase. And S phase stands for synthesis. And this is when your cells copy their DNA. Remember, their DNA is the cookbook that holds all their recipes. Genes are recipes that tell your bodies how to make proteins. And remember, you are you based on the proteins you make. Your stomach cells make a certain kind of protein, and that tells them, hey, you're a stomach cell. Your brain cells make a different kind of protein, and that says, hey, you're a brain protein. So when your cells are getting ready to make new cells, they have to photocopy every single gene. They have to make double the amount of DNA. So when they split in half, each cell has the equal recipes. Then they go through what we call the G2 phase. And the G2 phase is basically where the cell says, okay, have I done everything I'm supposed to do? Let's divide. So when the cells divide, this is what we call the M phase. And the M phase is mitosis and cytokinesis. Because cytokinesis actually happens about the same time as some of the end steps of mitosis. But the formal definition of mitosis is nuclear division. This is why we can't say bacteria do mitosis, because they don't have a nucleus. Bacteria do what's called binary fission. Binary fission, fission means to split, bi means into two. Process is kind of the same. They'll replicate their DNA, they'll grow to twice their size, and they'll divide in half. Okay. Well, remember with bacteria, they only have one piece of DNA to keep track of. We have lots of pieces of DNA. And remember, 99% of the time, our DNA is this tangled up mess. And this tangled up mess is called chromatin. So it's like having a sock drawer full of socks. Well, you dump your sock drawer out and you lay your socks out nice and neat so you can see what you have. At that point in time, our DNA gets organized. That's what we call chromatids. And then we put the matching socks together, and that's going to be what forms our chromosomes. So remember, chromatids and chromosomes only show up when the cell is in M phase, or it's getting ready to go through cell division. Towards the end of M phase, we'll have what's called cytokinesis. And the root word cyte means cell, to kinese means to split. So in cytokinesis, this is where the cytoplasm or the cell actually splits in half. So let's look at mitosis. Okay. Now, in the case of the cell cycle, please remember that interphase is not part of mitosis, but interphase is part of the cell cycle. Interphase is what we call the longest part of the cell cycle, due to the fact that this is where the cell spends most of its life. So interphase is the cell doing its normal job. But like I mentioned, if the cell realizes it needs to divide, it will go through certain stages. The first thing it's going to do, like I mentioned, is the G1 phase. And this right here would be the G1 phase. The cell's going to grow to twice its normal size. While it's doing that, it'll go through the S phase. And the S phase is where the chromatin duplicates. Remember, DNA in its what we call workable form is chromatin. And chromatin is this tangled up mess. However, chromatin is kept nice and neat in the nucleus. It's protected. So in the S phase, we actually take your DNA, which is a cookbook, and we photocopy every single page. So in the end, we're going to have two cookbooks that are identical copies of each other. And in animal cells, we have these things called centrioles. Centrioles are what I call the fishermen. I also call them the cowboys, because as you guys know, fishermen will throw out fishing line to try to grab something and reel it in. Or your cowboy might throw a lasso to catch an animal in order to bring it to him. Well, we have these things called centrioles that are going to throw out fibers. And these fibers are going to catch our chromosomes and help us move them around. So when the cell divides, we can make sure that everything is split up evenly. Now, the first step of mitosis is actually what we call prophase. Prophase is the longest part of mitosis because prophase is where the cell has to get organized. So think about your room. Think about a closet or your junk drawer. It's real easy to shove things in your closet. However, there comes a time where you have to get it organized. You have to take everything out. You have to try on what fits, what doesn't fit. You have to clean up. You have to get things organized. Well, that's what happens in prophase. In prophase, your cell needs to divide, and it needs to make sure that you have 2 of everything. And it needs to make sure that everything is nice and neat and organized. So prophase is the longest stage of mitosis because this is where the cell is getting organized. Well, the first thing that's going to happen, as you can see in this picture right here, is the nucleus is going to start to fall apart. Okay. So in prophase, your nucleus is going to start to fall apart because we have to move things around. So we have to get rid of that protective covering around our DNA. That is why this has to happen so fast, because when the nucleus disappears, our DNA is not protected anymore. This would be like you taking your sock drawer and dumping it out on your bed. And then you've got this tangled up mess of socks. Remember that tangled up mess is called chromatin. And then you can start organizing everything. You can start separating this tangled up mess into individual socks. I remember when we take our DNA from this tangled up mess and turn it into nice, neat pieces, we call those chromatids. But remember, we doubled our DNA. So now we have 2 of each. So that's like having 2 of each color sock. So once we untangle our chromatid into these nice individual pieces, we call those chromatids. Then we will stick the matching chromatids together. And when we stick the matching chromatids together, we'll connect them with a protein called the centromere. And that is what a chromosome is. So the only time we have chromatids and chromosomes is during cell division. Okay? The rest of the time when the cell is in interphase, our DNA is in the form that we call chromatin. So in prophase, our nucleus has to disappear. And that's to give room for the DNA to get organized. So it's like you taking your sock drawer and dumping it out on your bed, and then you lay out all the socks so you can see what you got. And then when you find your 2 green socks, you put them together. And that's going to be what forms the chromosome. Okay. Like I have here, the nucleus will disappear. And then the centrioles, my fishermen, are going to start to move to opposite ends. And so you can see that this is my centriole here. Here's another centriole. And they start to throw out their fishing lines. And their fishing lines are these things called spindle fibers. And that's what you're seeing forming in between them right here. Okay, those are my spindle fibers. And my centrioles are going to move to opposite ends. So I'm going to have 1 fisherman or 1 cowboy on each end of the cell. And they're going to start fishing. They're going to start throwing their fishing lines. And their fishing lines will start to attach to the chromosomes. So remember, I have a chromosome. And it's connected with a centromere. Okay. 1 fishing line will attach to this chromatid, and a different fishing line will attach to this chromatid. And together, they're going to start moving the chromosome to the center of the cell. So prophase takes a little while to happen because first you have to get everything organized. You have to get your nucleus to disappear. You have your chromatin has to become chromatids. Then you have to find their matching pairs to make chromosomes. You have to make these spindle fibers. The fishermen have to, the centrioles have to cast out the spindle fibers. They have to connect to the chromosomes. So prophase is the longest part of mitosis. And then everything starts to happen really quick. Once the centrioles have attached to the chromosomes, they're going to line everything up in the middle. And this is what we call metaphase. Now the root word meta means middle. So as you can see here, we have lined up all the chromosomes at the equator. And so right here is one of my centrioles. Here's my other centriole. And you can see how their fishing lines have attached to the chromosomes in order to make sure that they move. Now, at this point in time, the cell has to go really fast because the cell cannot make new proteins. So if the cell happens to run out of proteins in the middle of metaphase, the cell is going to die because it can't make new ones. Because remember, when DNA is in chromosome form, it doesn't work. So prophase, interphase is where the cell spends most of its life. So it's the longest part of the cell cycle. Prophase is the longest part of mitosis because you have to get everything organized and ready to go. Metaphase happens very quickly because everything's just going to line up here in the middle. Then, just like the fishermen start to reel in their fish, your centrioles are going to start to reel in those spindle fibers. And it's going to pull, it's going to pop the centromere. Remember, the centromere is the little protein right here that connects the two together. So one spindle fiber is going to pull one chromatid that way, and the other spindle fiber is going to pull the other chromatid that way, and it's going to pop the centromere. And therefore, our chromosomes get split in half. So, I remember they're copies of each other, so it's like having two page ones. So this is page one, and that's page one. So when the centromeres start to reel in their spindle fibers, this side of the cell is going to get page one of my cookbook, and this side of the cell is going to get page one of my cookbook. Now there is a mutation called non-disjunction, which we'll talk about more later, but non-disjunction is when anaphase, something goes wrong in anaphase, and a chromosome does not get split in half. So what would happen is, if I had a cell, what usually happens in anaphase, in non-disjunction, is the spindle fiber breaks. So here, these chromatids got split in half, but right here, this one did not. So it non-disjoined. Non meaning not, disjoin meaning coming apart. So it did not come apart. So what happens is one cell will end up with an extra chromosome, and that would be like something like Down syndrome. And the other cell ends up with no chromosome at all. And in most cases, that cell will die because it does not have all the DNA that it needs. Okay. But we'll talk more about non-disjunction when we get to genetics. But if non-disjunction happens, it's going to happen in anaphase. Then we have telophase. Some people say telophase. If you can remember prophase, this is pretty much the exact opposite. Anything that happened in prophase, the opposite happens in telophase. In prophase, the nucleus disappeared. In telophase, it's going to start to come back. But notice now, I've got two nuclei. In prophase, we made spindle fibers. Here, the spindle fibers are going to disappear. And in prophase, we went from chromatin to chromatids to chromosomes. That was prophase. Chromatin to chromatids to chromosomes. Here, we're going to go backwards. Here, we have chromatids. And so our chromatids are going to uncoil and become that tangled up mess again. So telophase is the opposite of prophase. Prophase, the nucleus disappears. Telophase, it comes back. Prophase, we make spindle fibers. Telophase, they break down. In prophase, we go from chromatin to chromatids to chromosomes. Here, we're going to get our tangled up mess. But as you can see, we're going to end up with one big cell with two nuclei. And inside that nuclei, they're going to have that tangled up mess. So usually during telophase is when cytokinesis starts. And you can see that in this picture right here. You can see that the cell is already starting to divide. Okay? And that's the process that we call cytokinesis, as you can see here. Now, depending on if you're a plant or animal, cell division does occur differently. This is an example of an animal cell. In an animal cell, we tend to pinch inward through cytokinesis, and we form what's called a cleavage furrow. So we start with the cell with two nuclei, and it starts to pinch inward until we have two cells. Okay. And where it pinches inward is what's called a cleavage furrow, as you can see right here. Plant cells form differently because plant cells have a cell wall. They cannot pinch inward. Okay. They're not as flexible. So therefore, what they have to do is form a new wall in the middle of them. So when the cell is done with telophase, notice it still has this cell wall around it. It's then going to go in with vesicles and create new bricks and form a new wall going right down the middle. And when we're finished, we're going to have two daughter cells that are identical to the parent cells. So in humans, we have enough DNA for 46 chromosomes. And let's say our cells are the size of a dime. In the end, I'm going to have two cells that are the size of a dime, and they're going to have enough DNA for 46 chromosomes. So they end up with making clones or exact copies of themselves.